Hello everyone and welcome to Anatomy. Today we're going to start with embryology. You may wonder why we start with embryology. Well, embryology is the basis of all anatomy really, and it kind of gives you the groundwork to be able to explain and understand things a little bit more thoroughly. And that's why I start out with the embryology aspect. So let's get started. The first thing I'd like to talk about is just kind of a bigger overview of timelines. So basically at the beginning we have a zygote. The zygote is when the sperm and egg nuclei collide. Those first 2-week period we call the blastocystic period, or it becomes a blastocyst. After the collision we get cell division, and that's where we get the blastocyst. At 3 to 8 weeks we have a period called embryo, or it becomes an embryo, I should say, not a period, but becomes an embryo. And then from 3 to 9 months it becomes a fetus. And then birth to 1 year we consider infant or infancy. Toddler is 1 to 3 years. A child is 3 years to, and I put a question mark because this really has varied over time. For example, an adult which is 18 plus can go into the military, but where does that child and adult line, where is it drawn? I can't exactly always say. It varies. But for this purpose, for the purpose of this class, anything 18 and older is considered an adult. So initially we get the zygote forms, and that would be right here at this location where the sperm and egg nuclei collide and the egg becomes fertilized. Then we start to get cell division. And eventually a ball of cells form, and this ball of cells is referred to as the morula. And that would be that object right there. And then as it develops more and more, we start to develop 2 cell divisions. And these 2 cell divisions consist of embryoblasts, which would be right here and right here, and then we have trophoblasts. The trophoblasts would be around the outside. Now what you're seeing here is basically a cross section of a uterus. So this is the uterus, and this is a cross section. In addition, these are all cross sections of the blastocyst as it's progressing through this first 2 weeks. So think of a basketball, and in the outside of that basketball is considered the trophoblast. Now their cells are a little ball of cells inside of that basketball, which we call the embryoblast. And those embryoblasts would be like sitting in the bottom or on the side of that basketball. Now if you cut the basketball in half, you're going to be able to see the outer layer of the trophoblast and then also this inner layer of the embryoblast. So there's our trophoblast. Now the trophoblasts eventually develop into the placenta and membranes as it embeds into the uterus, and the embryoblasts develop into the actual embryo itself. Now if I take this image, or actually better yet this image, and down here it's going to actually be the same image, however now it's embedded into the uterine wall, fully embedded. So out here in red we have the uterus. Okay. Now inside here we have 2 cell layers. We have the embryoblasts and we also have the trophoblasts around the outside. 2 cell layers. And we have a hollow spot in the middle, and we call this hollow spot the yolk sac. Okay? So that hollow spot is called the yolk sac. Again, the trophoblasts develop into the placenta and membranes, whereas the embryoblasts develop into the embryo. As we continue on in the timeline, we're going to find that we're at week 2 now, and the embryoblast is going to form a bilaminar disc. So right here, the embryoblast, which was that blue layer, is going to form a bilaminar disc. This bilaminar disc is going to consist of an epiblast layer and a trophoblast layer. I'm sorry, a hypoblast layer. Sorry, I misspoke. A hypoblast layer. Now, around the outside, we still have all of the trophoblasts. So now within this basketball, we have 2 layers of cells on the inside. We have an epiblast and a hypoblast. 2 cell layers, and that's where we form this, what we call a bilaminar disc, which means 2. Now, previously we labeled this the yolk sac, and it is still referred to as the yolk sac. But now we also have one more hollow area, and this hollow area is actually called the amniotic cavity. If you think about it, amniotic fluid is a very common term in pregnancy, and that's where the baby actually sits inside this fluid. And we'll kind of explain how this amniotic cavity eventually engulfs the entire structure or fetus. So over time, these epiblasts, which are here in blue, they're going to proliferate or divide. And as they proliferate and divide, they're going to turn into another type of cell. So they're going to basically grow into the space between the epiblast and the hypoblast and the trophoblast. Okay? So these are going to proliferate and continue to grow, and then they're going to migrate in between these layers. And what we end up getting is this area right here that layer on the outside, which we refer to as extraembryonic mesoderm. Okay, and this is between the epiblast and those in that hypoblastic layer. So a quick recap. Oh, let me finish coloring this. That'll be our trophoblast. So just a quick recap. We have basically fertilization of the egg. We start to get division of cells, and we develop a ball of cells called a blastocyst. Within this blastocyst, we have an embryoblast, right here, and trophoblast. So the embryoblasts are inside and the trophoblasts remain on the outside. Now those trophoblasts will remain on the outside. The embryoblast is what actually changes. So the embryoblast is then going to form a bilaminar disc. So the embryoblast becomes what we call epiblast and a hypoblast. So embryoblast basically divides and becomes those 2 cell layers, as you see here. We have a 2-layer disc in the middle, and this 2-layer disc is referred to as the bilaminar disc. As we continue onward, the epiblasts are going to differentiate and proliferate into another layer. This is called extraembryonic mesoderm, and these cells actually migrate between the epiblast and hypoblast layers and between the trophoblast layer in pink here, and that's the extra embryonic mesoderm. Excuse me. Next, the extra embryonic mesoderm, which was in pink on the previous slide, splits and forms the chorionic cavity. So let me draw that for you. So right here, we'd have that extra embryonic mesoderm. And that extra embryonic mesoderm actually splits, and that's what I'm drawing here. And it forms the chorionic cavity. So now we have three cavities. We have the chorionic cavity, we have the yolk sac, and we have the amniotic cavity. Now, remember, around the outside, what do we have? We have the trophoblast. And the bilaminar disc is still considered epiblast. Oh, wrong color. And hypoblast. Now, instead of concentrating on these three cavities, we're actually just going to look at this one right here. And that's what we're going to draw from now on, is just that segment. So what you're seeing here, based off of that previous picture, is around here we have our extra embryonic mesoderm. Okay. We have our epiblast. And we have our hypoblast. Now, at this week 3 to 8, we have a process called gastrulation, which happens. And this is when we actually form a trilaminar disc. So basically, we started out with embryoblasts. Embryoblasts differentiated into epiblasts and hypoblasts. And now to develop a bilaminar disc. Now we're going to develop a trilaminar disc, 3 layers. Now, basically what happens in this process is the epiblast cells invaginate into what we call the primitive streak. I'll tell you a little bit more about that in a minute. And they're going to replace the hypoblast to become the endoderm. So the hypoblasts actually get replaced by epiblasts. And this forms a new cell layer called mesoderm. And the epiblasts differentiate into ectoderm. The end result is a trilaminar disc consisting of ectoderm, mesoderm, and endoderm. And this will be where all of our tissues are derived from. So as you can see in this image, we have that epiblast layer. It's actually going to invaginate in, create a little valley, and then as that valley is created, it's going to replace the hypoblast, becoming the endoderm. So it comes in and replaces all of this in yellow and becomes the endoderm. technical difficulties. And then as we continue on, they form a new cell layer called the mesoderm, which will be here in red. So the epiblasts, again, invaginate in, replace the hypoblast to become the endoderm. Here they develop a layer between the epiblast and the hypoblast endoderm layer, and this is called mesoderm. Not extra embryonic mesoderm, but just mesoderm. Okay. And then these epiblasts actually differentiate themselves and become ectoderm. So now we have a three-layered cell. 1, 2, 3. Those three layers are ectoderm, mesoderm, and endoderm. And we have two cavities in this picture. We have our amniotic cavity, and then we have our yolk sac, and then all around the outside would be our chorionic cavity. I know it might seem a little confusing, but go back to this picture. We have our amniotic cavity, which is surrounded in blue, our yolk sac surrounded by yellow, and then our chorionic cavity surrounded in pink. Now, we're actually not going to look at anything outside this extraembryonic mesoderm, and we're going to basically just get rid of everything outside that chorionic cavity. And this is what it looks like. Okay, we got our extraembryonic mesoderm, then surrounded in blue is the amniotic cavity, surrounded by yellow is the yolk sac. Now remember, embryoblasts become the epiblasts, then the epiblasts differentiate to become three layers. They differentiate and become endoderm, mesoderm, and ectoderm. They become all three layers. Now we talked how the epiblasts actually invaginate, which is in that area right there. They invaginate into that what we call a primitive streak. So if you look at this next picture, remember we were describing everything as shaped like a basketball. Remember back to the morula, shaped round like a basketball. Now this basketball starts to form into kind of a football shape where we have two ends. We have a cranial end and a caudal end. If you think about a football, or you take a basketball and you actually elongate it into a football shape, that's basically here what's happening. The cranial would be one point and the caudal would be another point of the football. If you still cut that football in half, you would see, so if I was to cut the football directly in half and rotate it up, I would see three layers. Okay, I would still see here my ectoderm, my endoderm, and my mesoderm. Those would be the three layers creating a septum within the football. And one end of the football would be the cranial end and the other end would be the caudal end, meaning tail, meaning head, basically. Now, I've just simplified things by not drawing the blue around the amniotic cavity or the yellow around the yolk sac, just for simplicity's sake. And we'll go a little bit. This just basically is to show you the three layers, 1, 2, and 3 that happen. Now, as those epiblasts migrated into the primitive streak, right here we have that primitive streak. that primitive streak is where all of those cells migrated in. And what you're looking at basically here is, on this side, is you're looking at the football in an anterior to posterior cross section. So if I was to basically take the top off of this football and I was to look down into the football from the top, what I would actually see is this structure right here. Now if you look at my arrow going down here, draw it again, what color would I see if I cut off the top? I would see blue. So all of this here would be a blue color. And that was the epiblast, which eventually become ectoderm. Okay, but the epiblast, they migrate into this primitive streak, and as they migrate in, they go up towards the head, and then they also migrate down around the tail. Now there's 2 points here. Okay, there's 2 points. One is called the bucopharyngeal membrane, and the cloacal membrane. The bucopharyngeal membrane basically is where the mouth will be, and the cloacal is where the urogenital system is going to remain. So basically, if you think about your mouth is connected to your anus via a tube. No getting around it. That's just how it is. The bucopharyngeal membrane is one end of the tube, which would be the mouth, and the cloacal membrane is the other end of the tube. Now remember, in humans, our tube is actually divided into a fecal system and a urinary system. But if you think about a bird, they have one output, and that's where urine and fecal matter is mixed, and they have one open area. So, but in humans, it's actually split, and we have two. But that's what we'd refer to as the cloacal membrane. So the end result of all of this is a trilaminar disc. And this trilaminar disc, as I mentioned earlier, consists of ectoderm surrounding the amniotic cavity, mesoderm now differentiates into some more defined areas, and our endoderm. And again, here we have two cavities. We have the amniotic cavity and the yolk sac. Now I want to say a little something about the mesoderm here. The mesoderm is actually broken up into three regions. And those regions, so if I was to draw it out, it would look basically like this. Okay. So I've basically just drawn that and moved it to there. What we basically have here is we have three divisions. We have the paraxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. And we'll talk more specifics about that later. But then we have these two divisions of that lateral plate mesoderm. Somatopleuric, soma meaning body, and splanchnopleuric, splanchno meaning like gut tube, guts, mesoderm. So the somatopleuric is going to go along the ectoderm, on the ectoderm side, and this splanchnopleuric is going to go towards the endoderm side. So you can see that here, A and B. So let's go through each of these individual layers. The ectoderm, which I've colored in blue. The ectoderm is going to become our skin, sweat glands, sebaceous glands, and our enamel of our teeth, basically the outer covering of our body. Mesoderm is quite interesting. It's great. It's going to become our, especially this paraxial mesoderm, right here. So it's going to become our axial skeleton, voluntary musculature, so this would be like skeletal muscles, like the biceps brachii or the quadriceps, the dermis of just the back, and it's going to form basically 44 pairs of sommetomeres, which are basically compressed areas of this mesoderm, and you're going to have one on each side, one group on each side of the notochord. And this right here's the notochord, and you'd have two, basically two pair or two pieces of paraxial mesoderm. Now, if you think of a hot dog or a sausage, and you take a tube and you fill that tube, or better yet, let's think of a balloon, one of those long clown balloons. Think of that as being a column of mesoderm. Now, if I take that column or that balloon full of mesoderm and I start twisting it, I start to develop links within that mesoderm, or small compact vessels of mesoderm. So basically, if I had this long balloon, and then within that balloon I started twisting it, it would start to look like this. And then if this was midline, I'd have that on both sides of midline, going all the way down. And that's what you're seeing right here, is that paraxial mesoderm going down each side of midline, or on each side of the notochord. Okay? And each one of those we call a sommetomere. Eventually they'll become somites, but right now we're just going to refer to them as sommetomeres. So remember, if this is considered midline, and we have the bucopharyngeal membrane and the cloacal membrane, one at the head or cranial end and one at the tail or caudal end. Now on each side of these, remember we talked about having that column of mesoderm, and then that column breaks up into individual units, like twisting a balloon. And basically you get these little balls of mesoderm on both sides, and there's 37 pairs of them on each side. The first 7 of the 1 through 7, these are actually going to go to the striatum muscles of face, jaw, and throat, and they're associated with what we call pharyngeal arches or branchial arches. For the purpose of this class, we are not going to go into the pharyngeal arches. So those first 7 are basically, we're dismissing as centimeters going to the head and neck. We're not going to talk about specifics. Now 1 through 4, which would actually be 8 through 11. But now, if you look here and we draw these lines over, after we get rid of the centimeters, or they migrate to the head, we then call everything else somites, or Semitic musculature. It would be, I'm sure you maybe have heard of. So they're a 1-to-1 ratio. It's just before they were centimeters, and then the first 7 go up to the head, and then the remaining ones, which are, and I should step back. With the centimeters, there's actually 44 pairs, and then with the somites, there's 37 pairs. I apologize for that. And you can see it right here. I think I may have said that incorrectly before. So centimeters, there's 44 pairs. Somites, there's 37 pairs. The first 4, okay, are going to contribute to the base of the skull, basically. The occipital part of the skull and bones around the eyes and inner ears, extrinsic ocular muscles, and muscles of the tongue. Those are going to be the first 4 somites, or centimeter 8 through 11. Now we have the next 8, which are going to contribute to cervical vertebrae and associated muscles and cervical back dermis. Okay. So if you think about it, this number 8, each one of these would be associated with some form of neural tissue. And how many cervical nerves do we have? We have 8. Okay. Here, 13 through 24, 13 through 24 somites. These are going to contribute to the thoracic vertebrae. Remember, axial skeleton, the paraxial mesoderm contributes to axial skeleton, and the associated muscles and part of the abdominal wall, upper limb musculature, and thoracic back dermis. Okay, so look at this, upper limb musculature. So axial skeleton, and then our upper extremity musculature, and then a little bit of the dermis on the back. So that's going to be 13 through 24. Now if you think about this, 13 through 24, that's 12 balls of tissue, or 12 somites. Each one of those is going to have a nerve going to it. Well, how many nerves do we have? 12. In that thoracic region. Now as we go through 25 to 29, these are going to be associated with the lumbar vertebrae, the lumbar musculature, and some of the abdominal muscles, and then also the lower limb muscles. And lumbar, 25 through 29, there's actually 5 of them, 5 balls of somites, or 5 somites, which are balls of tissue. Each one has a neural component, and therefore we have 5 lumbar nerves. The same thing happens for the sacrum. Okay? And then for the coccyx, we actually typically have 1 to 3 coccygeal vertebrae, and we only have 1 nerve going to that typically, and that would be coccygeal 1 nerve. So only one of the nerves is innervated by tissue coming from neural tissue. But they actually think potentially that in this region, those last 2 somites, even though they don't get direct neural input, the phylum terminale that extends down to the base of the spinal column would be equivalent to nerves coming out, or the epineurium of the nerves coming out into the somite 36 and 37. So I know that's a lot to take in. I will be happy to explain it during lecture tomorrow a little bit more thoroughly, or I should say with our discussion tomorrow. All right, moving on. So we talked about ectoderm, we've talked about paraxial mesoderm, and just as a quick recap, paraxial mesoderm becomes the axial skeleton, voluntary musculature. So think about it, biceps, triceps, quads, anything you can voluntarily contract below the head and neck. Also becomes the dermis of the back, and we talked about how it forms somites. Now we're going to next talk about this intermediate mesoderm. The intermediate mesoderm actually forms the urinary system and part of the genital system. So if you think about your urinary system, what is one of the main components of it that you need to be aware of? That would be your kidneys, right? Well, are your kidneys located behind your gut tube? Absolutely, right? They're in the posterior part of the abdominal wall. And we'll talk about that in a little bit more detail later. Next, for the mesoderm, we come to the lateral plate mesoderm. Now remember, there's two divisions of this lateral plate mesoderm, A and B here. A goes along with the ectoderm, and which forms our skin, so it makes sense of the body or the body wall. And that's going to be the soma or somatopleuric mesoderm. It gives rise, the actual somatopleuric mesoderm gives rise to the inner linings of the body wall, which is dermis, which is the remaining parts of the trunk, and the limbs. Okay, so all the dermis of the limbs is formed by this somatopleuric mesoderm. And it also forms all the bones and connective tissue of the limbs. Remember, the paraxial mesoderm, or the somite, developed our axial skeleton, whereas our appendicular skeleton is formed by the lateral plate mesoderm. Now the splanchnopleuric mesoderm, okay, B, is going to be adjacent to the endoderm. So here we can see the endoderm. And it's going to be adjacent to it. So as it migrates out, it's going to give rise to the coverings of the visceral organs. So if you think back, you have parietal pleura and visceral pleura. Parietal pleura, which is on the outside or lining the body wall, that would be derived from somatopleuric mesoderm. The visceral pleura, which is stuck to the organs, would be derived from splanchnopleuric mesoderm. All right, moving on. Now we get into the endoderm. The endoderm, in yellow here, this is going to become the digestive system, the respiratory system, including the liver. So if you think about a tube that runs from your mouth to your anus, and off that tube you're going to basically develop other organs. But all of those organs seem to connect in one way or another. Just to give you kind of an example, if you have a tube, and think of that tube as being kind of your throat and basically your bronchi or your trachea and your esophagus, okay, combined. You just have one tube. Well, that tube actually develops a septum and splits. So instead of being one tube now, it develops into two tubes. The trachea then splits and develops into lungs, okay, where the esophagus continues down and it develops a bulge which becomes the stomach. Then it continues down into the duodenum where we have a duct coming off of the liver. So the liver was developed from the endoderm, and basically all of it's connected where we have enzymes which dump from the liver into the gut tube. Okay? Same thing with the pancreas. We have basically the major and minor papilla which dump in fluid into the gut tube or into the intestine. Then that intestine travels all the way down and eventually arrives at the anus. Now, remember, the mouth would be equivalent to the bucopharyngeal membrane, also the nasal cavities and the opening for the nose and all of that. But just for simplicity, think of it as the mouth. And that cloacal membrane would be, think of it as being your anus for simplicity, but realize in some animals like birds, they don't have a separate urethra and colon. They're combined into what we call a cloacal membrane, or into a cloacum. Sorry. Sometimes words are hard to say. So that's basically how the gut tube runs. Now we're going to talk about a process called neurulation, and this is how we develop our spinal cord. And if you remember back a few slides, we have the 3 layers of the trilaminar disc, which include ectoderm, mesoderm, and endoderm. And that's what you're seeing right here. We have our ectoderm, our paraxial, intermediate, and lateral plate mesoderm. Paraxial, intermediate, and lateral plate mesoderm, specifically somatopluric and splenopluric. And then we have our endoderm. Right now, I want to concentrate just on the ectoderm. So in this ectodermal layer, which would be basically right here, we're going to get another invagination. And you can see here how it invaginates right there. And it develops 2 specialized points on each apex of that invagination. And you can see as it invaginates more and more, we get these specialty cells located there. We have the ectoderm here. And the ectoderm actually starts to develop into a different type of tissue, and these 2 are actually going to connect. So when they connect, what we end up getting is 3 layers. We get the ectoderm, we get this specialized layer of ectoderm called, it's eventually going to be your neural crest cells. And then here we have another layer called the neural tube. So we have ectoderm, neural crest, and neural tube. Now realize this structure is running from cranial to caudal. Okay? Running cranial to caudal. So in this picture right here, you can see how we have the ectoderm, those 2 will eventually come together, and then down below these 2 will come together and we'll get a neural tube. And then over the top we have a small layer of specialized cells. It's a little bit bigger than I wanted. There would be our neural tube. So we basically get our 3 layers. Our ectoderm, neural crest, and neural tube. Now that neural tube becomes your brain, your spinal cord, your skeletal motor neurons, your preganglionic neurons, and your inner neurons. We'll talk a little bit more about these specifically when we talk about the autonomic nervous system along with the central and peripheral nervous system. But my question for you is, this is a tube, right? You can see how it's hollow. Is our brain hollow? Yes, absolutely. Why is our brain hollow? Well, we have ventricles. So you might ask, is our spinal cord hollow? Well, it is down into the cervical vertebrae where we have the central canal. I should say into the cervical cord. But then eventually that hollowness pinches off, or as the outer structures continue to multiply, it basically pinches off that inner tube. So as the spinal cord goes down below the cervical region, it becomes a potential space. You can't actually see the space anymore, but that's referred to as the central canal. Now, what do skeletal motor neurons do? Well, these are going to innervate those somites, right? This is going to provide all of our voluntary musculature. And then we'll talk a little bit more about the others later. Neural crest, look at where they're located. Right on the dorsal aspect of the spinal cord. What else do we know from our undergraduate anatomy that's located right there? Well, we know that the dorsal root ganglion are located in that spot. And so the neural crests actually become all the sensory neurons of the body. If you remember back to your undergrad anatomy, you should have had dorsal root ganglion. That's where all the sensory cells come in. We'll talk more about it later, so don't get too worried. The neural crest also contribute to brachial arches, which is an FYI. That means for your information. I will not test you on that. I just think it's important that you know it. Well, just important that you realize what's happening. And the connective tissue of the head is also derived from neural crest, which is also an FYI. However, the second motor neurons of the autonomic nervous system and the medulla of the adrenal gland, you will have to know, are derived from neural crest. So all of these are very important. So during this embryonic period of weeks 3 through 8, we have a lot going on. We get modification of this trilaminar disc by this lateral body wall folding. From this is how we basically get our body layout. Because right now, if we look at this picture right here, and we consider this as being our ectoderm. Well, we know our ectoderm makes up our skin. Here we have our, I'm going to see if I can get a yellow pen. Here we have our endoderm. That endoderm is going to develop our gut tube. And then here we have our paraxial mesoderm, our intermediate mesoderm, and our lateral plate mesoderm. Okay? Now on top, on the dorsal aspect, I shouldn't say on top, I should say dorsal aspect, we know this is the amniotic cavity. Now, if we think about the whole pregnancy component of being within the womb, what is the baby or the fetus wrapped in? Well, it's wrapped in this amniotic cavity. Well, how does that happen if the amniotic cavity is only on the dorsal aspect? So I think, from my mind, to think about this body folding, if you follow the amniotic cavity, I think it helps make a little bit more sense. So we'll go through a few examples of that. As this lateral wall begins to fold, you can see that the ectoderm starts to migrate more ventrally, and with that it's dragging the paraxial mesoderm and the lateral plate mesoderm and the intermediate mesoderm. So really, truly, it's dragging that lateral plate mesoderm out quite a bit. And then we have our endoderm. As this continues to migrate or fold more anterior, that ectoderm begins to fold around more so. The lateral plate mesoderm continues to migrate. And our endoderm remains in the middle. And we still have our paraxial mesoderm and our intermediate mesoderm and our amniotic cavity. And you can see that amniotic cavity starting to wrap around the structure. So as we go to this image, the ectoderm continues to fold. Okay. We have our lateral plate mesoderm, specifically that somatopleuroid going with the body wall, wrapping around. And then we have our splanchnopleuroid, which is along the gut tube, which the gut tube we know is derived from endoderm. So at some point here, this whole gut tube is going to migrate internally. The two ectoderms are going to come together and the two mesoderms are going to come together. The two somatopleuroid mesoderms. So what we end up having around the entire outside is our ectoderm. Internally, we have our somatopleuroid mesoderm, which is part of that lateral plate mesoderm. We have our splanchnopleuroid mesoderm, wrapping around the endoderm or the gut tube. In addition, we have here the somatopleuroid or the somites, the somatic mesoderm, and then we have the intermediate mesoderm. And in the center, we have our neural tube. And then on each side, we have specialized cells, which we call neural crest. I'm not worried about any of the other structures associated there. Just realize, for your own information, that structure right there is the aorta, running right down the midline. So eventually, this is what it looks like. Around the outside, we have our amniotic cavity. We have our ectoderm wrapping all the way around. We have our splanchnopleuroid mesoderm, which will be wrapping around the gut tube, or specifically around the endoderm. And we have the somatopleuroid mesoderm wrapping, soma meaning body wall. It's wrapping around the body wall. You may ask me, well, where is this exact division between somatopleuroid and splanchnopleuroid? No idea. We know it's in the posterior wall, but for me to say, oh, that's the exact location, I'd be lying to you. So from this picture, though, if you think about that splenopleuric and somatopleuric mesoderm and what you know already from the endoderm making the lungs, also making the liver and the gut tube or the intestines, we know that there's basically two layers within that thoracic and abdominal mesothelial layers within the thoracic and abdominal cavity. Those would be the visceral and parietal pleura around the lungs. So the visceral pleura, what do you think it would be derived from? Well, we know the lungs are derived from endoderm, so it'd be right next to the endoderm. So it's derived from splenopleuric mesoderm. We know that the parietal pleura lines the thoracic cavity, which is right near the body wall. So that is derived from the somatopleuric mesoderm. The parietal peritoneum is derived from somatopleuric mesoderm. Well, the visceral peritoneum is derived from, which is next to the organ, splenopleuric mesoderm. Pretty interesting, if you ask me. Next, we have the intermediate mesoderm. Now, where are the kidneys located? Well, if you think about it, the kidneys are located in that posterior abdominal wall, and they're actually behind the parietal peritoneum. So, with that said, they are retroperitoneal organs. They're behind this splenopleuric mesoderm or somatopleuric mesoderm, wherever the division is. Next, we have our dorsal mesentery, and this will become important when we talk about our viscera. But right here, this is our dorsal mesentery. Now, this is where all of our blood vessels come from the aorta and supply blood into the gut tube. And to give you an idea, if you remember from your undergrad, this would be like the superior mesenteric artery would run through there, or the inferior mesenteric, splenic, or I should say the actual celiac trunk and associated branches. Next, we have the neural tube. Neural tube was derived from ectoderm, and that becomes your brain and spinal cord. We have neural crest. Those neural crests are on each side, and those will develop into all the sensory neurons of the body, and then also second order neurons of the autonomic nervous system, which we'll talk about, or postganglionic neurons, I should say. Then we have these sclerotome, dermatome, and myotome. These are all derived from that paraxial mesoderm or the somite. Okay? And that's what you're seeing right here is the somite. So on the opposite side, I have it broken up into its different divisions. And if I enlarge the picture a little bit here and draw it in an enlarged view, it's basically divided up into three major divisions. And those three major divisions would be the dermatome, which is 1, the myotome, which is 2, and the sclerotome, which is 3. So you can see it here, 1, 2, and 3. Now let's talk about the dermatome. When we think about dermatome, what do we think about? Sensory innervation of the skin. So kind of keep that in the back of your head. Sclerotome. Sclero means hard, right? So think of that as being the axial skeleton, right? Because the somite makes up that axial skeleton. And the myotome, myo meaning muscle, Well, the somite makes up all of the voluntary musculature of the body, and that's where the somite is. But if we think even a little bit deeper, when we think about the innervation of voluntary musculature, there's really two types of innervation. There's a ventral rami and a dorsal rami. The dorsal rami goes to all of our back musculature, right? We call those the epaxial muscles, or the back. The ventral rami goes to all of our hypaxial muscles, or basically anything innervated in the anterior trunk and limbs. So here we have our epimere, which is A and B. Epimere is A and B. I'm sorry, epimere is A, hypomere is B. So epimere, well, let's step back. If we think about the sclerotome, it's going to basically migrate around the spinal neural tube and become our skeleton. Our myotome is going to migrate to A, will go posteriorly and develop the erector spinae muscles and the transversospinalis group muscles, all innervated by dorsal rami. And 2 is going to migrate to B, or the hypomere is going to migrate ventrally, and it's going to contribute to all the anterior trunk and limb musculature. And then the sclerotome, we'll talk a little, the dermatome is also going to migrate ventrally and dorsally and become the sensory component of our ventral and dorsal rami. So just talking a little bit about neural tube organization. Right here we have the neural crest, and yes, they do split here. So you get them on each side. And then we have our neural tube. Now the neural tube is derived into a dorsal or an alar plate and a ventral basal plate. Now if you think about the typical pattern that we see in the spinal cord cross section, that's what a cross section of a spinal cord would look like. And we know the sensory neurons are derived from neural crest. So basically we have a central axon growing into the cord, and we have a peripheral axon going out to the body wall, either to the back or ventrally. And let me actually draw it in a different color. Here's our neural crest. We know neural crest is going to develop sensory neurons of the body, so I'm going to draw them in blue. That would be a sensory neuron. We would have our central axon going into the cord, and we'd have our peripheral axon either going out a dorsal ramus or a ventral ramus to provide sensory information. Okay? But where is the cell body for sensory? Neural crest. Now, when we talk about all the voluntary musculature, innervation for all the voluntary musculature, those cell bodies are actually in the neural tube, and then the axon will either go out the ventral, or the dorsal, or the ventral ramus, and follow the somite out, either to become innervate epaxial or hypaxial musculature. So in this picture, dorsally is sensory, and ventral is motor. Now realize that this is called a dorsal rami, ventral rami, dorsal root, ventral root, and this right here is the spinal nerve. So if I was to cut the ventral root, what would I lose? Voluntary motor. If I was to cut the dorsal root, what would I lose? Sensory innervation, but no motor. If I cut the spinal nerve, I would lose motor and sensory from both my epaxial and hypaxial musculature and my dorsal and ventral body wall. If I cut just the ventral rami, I would lose motor and sensory from my anterior trunk and limbs. In dorsal rami, I would lose motor and sensory to my back musculature and skin in my back. So now I'm going to talk in a little bit more detail. So again, if we think about that kind of pattern of the spinal cord, which I just drew, so this would be dorsal and ventral of the neural tube. That's a very bad drawing. Let me redo that. So we have our neural tube. In that neural tube, we have gray and white matter. I'm going to just draw that in right now. Okay? And basically I have dorsal and ventral aspects of our spinal cord. So again, this is a cross section. That's where the central canal would be if it was there. Now on the dorsal aspect, we have a dorsal root ganglion. And that dorsal root ganglion is derived from neural crest cells. And it has a central axon and a peripheral axon. Now realize that there's thousands of axons going centrally and peripherally. I'm just drawing one. And where it splits here, it actually doesn't really split. There'd be two neurons. I just draw it as one for simplicity. And then here, our motor neuron would be there, and it'd either go out the ventral or the dorsal ramus to the back, or ventral musculature, or the epimere and hypomere, depending on how you want to look at it. Now, realize here, a little bit of test. A little test here, see if you've been studying. What is that structure? Spinal nerve. Here, ventral ramus. Kidding. That's the dorsal ramus. Just testing you. Here, ventral ramus. Here, dorsal root. And then here, ventral root. The spinal nerve, of course, as we said earlier. Now, if I apply this pattern to the image over here, this is what it looks like. Dorsal root ganglion, we would have our cell body for our sensory neurons. We'd have a central axon going into the neural tube, and we'd have a peripheral axon, which would follow the dermatome either ventrally or dorsally. We would have a motor neuron, and we know that voluntary motor neurons are derived from neural tube. Then that would migrate, and it would either go out the hypomere or the epimere, or migrate with on that somite to become the ventral trunk and limb musculature or the dorsal back musculature. So that is why we get that dorsal ramus and ventral ramus innervating two different areas of skin and two different motor areas of the body. Pretty cool if you ask me. Now when we talk a little bit about autonomics, it gets a little bit more complicated, and we will revisit this. But I just want to start the conversation now. And something you guys have to remember during this class is our first test isn't for a long time. I don't expect you to understand this first lecture fully after listening to it once. We're going to come back and revisit lectures all the time. I will do my best to make sure you are adequately prepared for that first exam and know the information. So don't stress out too much if you don't fully understand everything in this lecture. It will all come together in the end. And I will do, well, I can't say it will, but I'll do my best to help you get it so it will all come together in the end. We've had a pretty good success rate so far, and I'm not going to stop now. So with that said, moving forward, this might be new information for some of you, and others you may already have had this information, so just bear with me. So if I step back and I look at what is derived from neural crust. Neural crust contributes to sensory neurons of the body. We drew this, right? That's the dorsal root ganglion. It also becomes the second motor neurons of the autonomic nervous system. Now this is where I need you to think really strongly. Second motor neuron of the autonomic nervous system. So let me go back to this picture. Now this gets exciting. I hope you guys are getting excited. I am. So when we talk about somatic motor or voluntary musculature, we have a motor neuron which is in the neural tube, okay? So it'd be within that neural tube. And it's going to travel out and it's going to go to, let's say, the biceps. There's one neuron and one axon. Now, I realize there's thousands of them going to the biceps, right? But we're drawing it as a single one. But realize that from neuron to muscle, neuronal cell body to muscle, there's no break. There's no additional communication. Okay? So that's for voluntary motor. Okay? Now, what other motor system do we have? Well, we have our autonomics. Okay? Our autonomic system consists of sympathetic and parasympathetics. Okay? Sympathetics, fight or flight. Parasympathetics, rest or digest. It's not totally critical right now. But what is critical is that it's still a motor system, but now it's a neuronal cell body system. So we have a cell body in the spinal cord, which is going to go out, and it's going to synapse, and then go to an organ. Okay? So we have 1, 2 cell bodies. Let me do this. We have 1 and 2 cell bodies. Now, the same thing happens for sympathetics and parasympathetics. The lengths are just a little bit different. So I ask you, which one is sympathetic and which one's parasympathetic? Sympathetics have short preganglionic and long postganglionic. Okay? So this one here would be sympathetic. Whereas parasympathetics have short postganglionic and long preganglionic. Okay? and those are considered parasympathetic. So you might ask me, where are these ganglion located? Here's one, and here's one. Well, the ganglion right here, that one for parasympathetics is going to be really close to the organ, or sometimes even in the organ, whereas the one for sympathetics is typically within the parasympathetic chain and ganglion, right along the spinal column. Not always, but that'll give us kind of a little bit of insight. The other location that the sympathetics can be is what we call prevertebral. And I'll show you a picture of those, but that would be similar to your superior and inferior mesenteric ganglion. So, in the picture to the right, our neural crest cells are here. Okay, those neural crest cells migrate, and they become the dorsal root ganglion. They become paravertebral ganglion. Okay. Those paravertebral ganglion would be the same as your sympathetic chain and ganglia right along the spinal cord. And they also become prevertebral ganglion. So this is where your splanchnic nerves would go below the diaphragm. These are all, these two here, are sympathetics. Also, those neural crest cells will migrate down really close to the organ. These become the parasympathetic ganglion. Okay. So all of these are migrations from the neural crest. So neural crest become a few things, a few different ganglion. They become the dorsal root ganglion. They become the paravertebral ganglion for the sympathetic system. They become the prevertebral ganglion for the sympathetic system. And they become the parasympathetic ganglion that is close to the organ for the parasympathetic system. So basically, that is why we have all these ganglion throughout the body. These ganglion are not derived from neural tube, rather they're derived from neural crest, and they can migrate to different parts of the body. The last thing I want to mention is that back here, when we talked about what the neural crest becomes, we mentioned it also becomes the medulla of the adrenal gland. So basically, the adrenal gland controls, via the delivering, I should say it helps control the sympathetic system, the adrenal gland does. And it does that through this. Basically what happens is the neural crest cell that would become the secondary neuron, or the second neuron of the autonomic nervous system for the adrenal gland, the actual neural crest cell becomes the medulla of the adrenal gland. So it's interesting. The medulla of the adrenal gland, even though it's an autonomic system, it only has the primary neuron going to it, or that first order neuron, because the second order neuron is actually the organ, the medulla of the adrenal gland, which I think is pretty interesting, and I can talk to you more about that later. So with that said, we have now finished our first lecture. Again, I want to reiterate, I am not going to test you on this tomorrow. Okay? This is for you to listen, get an understanding of the words, kind of get some questions, and then we're going to go over it and over it and over it until you get it. Each lecture relates back to the autonomic nervous system because this is where, or back to the embryological system because this is where everything is derived from. So if you're feeling like, wow, this is too much, I'm overwhelmed, don't. Give it a little chance, let it sink in, and we'll go from there. You're in grad school. We all want you to finish. We all want you to do well. There aren't any classes where we weed anybody out. We all want you here, and we'll do everything we can to keep you here. So bear with us in this time, and I will talk to you more about it during our class time. Take care.